Hello students, welcome to Swayam MOOCs, and this course is on analytical techniques. The topic we are going to do today is on colorimetry and UV visible absorption spectrophotometry. Now this class is the second class in the week 1. So in the previous class I have explained to you the basics of the aqueous solutions, buffers, how important is pH in preparing solutions, because for any experiment you are going to do you need to have a dependable pH and the concentration. So having done that, now what we are going to do today is an extension. The two techniques, that is a colorimetry and absorption spectrophotometry, are used to determine the concentration of your interested molecule, that is either it could be a protein or it could be a protein, nucleic acid, or any other molecule. So these two are most widely used. I think it is one of the beginning experiments you do when you determine when you want to determine the concentration of a protein or a nucleic acid or even a drug or a dye or any molecule that gives a color or it can absorb in the UV visible region. So that is why the name suggests that colorimetry, you are determining the concentration of the molecule of your substance or an unknown in the visible range, which gives color. So as the name suggests, it's very simple. Now the second technique is a little more advanced, which also involves the visible range, that is it can be a colored compound or it can also absorb in the UV range. Now when a molecule absorbs in the UV range, you cannot actually determine the concentration using colorimetry. So colorimetry has its own limitations. So what you are going to do is in the UV range, you will use the absorption spectrophotometry. So basically both the techniques are important to determine the concentration, whether it is in UV range or in the visible range. Now having said that, colorimetry is routinely used or given to even to the UG classes where it is not very expensive, so it's very simple, not very difficult to use. However, when UV visible absorption spectrometry also can give, then why you can ask me why you use UV visible, even a visible range, why should you use a spectrophotometer? Because the spectrophotometer also can give you quantitative estimations. Nevertheless, it has much more advantages where you can determine the structure of molecules also, conformations of proteins and nucleic acids and so on. So the spectrophotometry is much more expensive, and of course it is much more sensitive, much more information it can provide. So all these techniques you can be used, whether it could be any substrate, enzymes or any other molecule. So when you talk about any spectroscopy, the name, remember the technique is called spectrophotometry. So whenever there is a term used, it is actually, the principle is based on the spectroscopy. Now what is a spectroscopy? That you are studying the molecular and atomic structure of a substance in observation, how it is interacting with the light or the radiation. Now when we talk about radiation, you must remember that any radiation can be ionizing radiation or a non-ionizing radiation. So we are only limited to ourselves with the non-ionizing radiation, which is the electromagnetic radiation. Now electromagnetic radiation, using this the basic principle is that absorption or absorbing the light in the range of electromagnetic radiation, that any spectroscopy can be used depending on the range you are using, the range of the EMR that you are using. So the techniques actually gives a lot of applications both in biochemistry, biotechnology, medical chemistry, molecular biology, and of course structural biology, where you understand the molecular structure of proteins and nucleic acids. And of course you can determine in the protein engineering, even it has applications in forensic science, bioorganic chemistry and so on. So when we talk about this electromagnetic spectrum, you should know that we are only limited to the UV visible. This particular class is only limited to the UV visible absorption range. Now what is electromagnetic radiation? In this range, you can see that radio waves, it has a different energy, microwaves have a different energy, infrared, then comes the visible, then of course UV visible, then X-rays and gamma rays and so on. So of course gamma rays or more ionizing radiation, we are not interested. So when you go from left to right, what you are seeing is you can arrange these the different kinds of radiation in EMR, that is electromagnetic spectrum, is that one is the wavelength and the other is the energy. So both are inversely proportional. So as you see, the energy of the radio waves is less as goes to the gamma rays, highest energy. Similarly, the wavelength, even in the monitor you can see the spectrum is clearly showing that the wavelength is higher on the left to the right when you have the less wavelength and high energy. So this is the basic fundamental thing. I'm sure you all of you know, because this is even you must have studied in the in your UG class. So the visible spectrum, which is coming to the first class, is that the colorimetry. So visible spectroscopy, you know that the white light is divided into various ranges of the spectrum, as you can see in the picture. So basically the violet light it has a wavelength of around 300 to 450, and similarly if you go to the extreme range, you have 600 to 700, that is very close to the visible range, that is has a bluish green. So this is the spectrum. Lowest is the red color, then of course is the VIS colors you can see. Now coming to the electromagnetic spectrum, that interaction with the matter, with the radiation, that is a fundamental thing. So most of us know that many different ways the transmitting energy and the phenomena come together in one physical entity, that is called the electromagnetic spectrum, and differences between these sources of radiation actually gives a different amount of energies. So this actually shows, the next slide shows the electromagnetic spectrum in terms of the molecular interactions that each molecule absorbs in a different wavelength, and therefore depending on their molecular structure. So each molecule has a different, each bond has a different energy, and therefore depending on its energy, if it is in the same range frequency, then absorption occurs. So atoms and molecules undergo electromagnetic transitions, whether it is in a visible or in the UV range. And then remember that absorption also we call it as transmittance or reflection, because what the molecule has already absorbed, you are not able to measure, because the energy has already been absorbed by a molecule. So what you can do, what you can determine or the measure is the property of that is not absorbed or transmitted. So in the whole thing, in the whole two UV visible absorption spectrometry and colorimetry, what you are measuring is the light that is not absorbed. When you give an incident light eye, what is not absorbed, that is the transmitted light you are going to measure. So this is something which is very important to understand. So based on this, the objectives are, the objectives I have laid out for this class are to understand the basic principle of electromagnetic radiation and how light is absorbed by the molecules. Then the most fundamental, most useful equation, that is a Beer-Lambert's equation, how to derive it and what is its application. Then comes the colorimetry in a more detail, more in a technical as well as the experimental details, and how to determine the concentration of a sample. I'll give you an example. Next, we'll do the absorption spectroscopy of proteins with an example. Of course, I'll give the introduction to the UV visible spectrophotometry. Then we will do the spectroscopy of the proteins. And in a protein, what is a peptide bond, what is absorbing, and then how are the aromatic amino acids and so on. Also the prosthetic groups. Now the conformation studies of proteins like denaturation also you can study, and absorption spectroscopy of the nucleic acids. And then the thirdly, the estimation of the concentration of RNA or DNA. Again, it is the most fundamental application you use every day in molecular biology when you determine a sample, that whether it is sample is pure or not. When you give an energy, because you are giving a radiation, E equal delta E is the difference in the states. Then if you give that much energy exactly between the ground state and that excited state, the molecule will absorb. So it is not that every molecule will absorb in all the range. It has its own natural frequency, natural energy, and that is what is shown in the picture, that E1 is a ground state, and if the energy states between a ground and excited is E equal to h nu, where h is the Planck's constant and nu is the frequency, then that molecule will absorb. So all the spectroscopic techniques are basically based on this absorption or the transmission of light, whether you can determine, but basically absorption. Now another one depends on the energy levels within the system. Therefore, the difference in the energy levels has to match the energy that is given to the molecule. So now the slide shows the colorimeter, which is a simple colorimeter which is used, that is the basic parts I am showing. On the left side hand you see that when you give a light, any light of one particular wavelength, that is monochromatic light, that intensity of that light is I0, the total intensity, and then you keep one sample of your molecule of your interest, that we call it as the concentration of C with some concentration, and then what you do is the light that is coming out of the sample, this light is thrown on a particular you call it a cuvette or any sample tube, and then the light that comes out, that is the energy that is been coming out or the transmitted. So that becomes, because some amount of energy has already been absorbed, so the I0 is a total and that is given out is I, that is less than I0. So these are the basic parts of the colorimeter. Now how does a colorimeter works? There is a source of lamp, that is a tungsten lamp, that passes the light through a small slit, that is called a filter. Then the slit is where a small amount of light goes and then falls on the sample tube, that is you keep it in a cuvette, and then of course filter the unwanted light, and then that falls on the photocell. What does this photocell do is to convert the light into a measurable quantity, because it has to be converted into electronic energy, electrical signal, and then of course you measure, that gives into the galvanometer, it gives a deflection, and so the needle moves. So basically you are converting the transmitted light by the sample to electrical signal where you can measure, and that signal of course is converted to connected to a galvanometer. So this is the basic fundamental principle. So all samples, any sample what you are measure dissolved in a solution has to be corrected for the background. Now therefore what you do is to require sap to subtract the background, whether it could be a water, it could be a buffer, or it could be any solvent that you are using, non-aqueous solutions, that has to be subtracted. So when a solution that contains all the components, that is buffer, except the one you are measuring the compound, that is called the blank. Now this we call reference, you can also call blank. Now then what you do is to adjust the reading to 0 if there is any kind of what do you call the scattering. So whatever light is scattered, that will be corrected into 0, and then you put your sample. I am giving you an example where you can determine the concentration of B12 using a colorimeter. B12 is a water soluble vitamin, and here what you make it is converted into a colored complex, and then the maximum at wavelength at which you measure is the 435 nanometer, which is actually complex with the cobalt ion and nitroso or salt. So this gives a colored complex. So what you do here is, as I shown in this small video, that you have a blank, then you have 1, 2, 3, 4, 5 the concentrations of the vitamin B whose concentrations are known but at a different concentrations. Then finally you have on the right extreme, you have an unknown sample for which you want to know the concentration. So you need to generate a standard curve. So now what you do is to add the vitamin, and then of course in the unknown sample you have add about 5 ml, whereas in case of the all unknown all other samples, you equivalent amount of water you add. So that way you will you were equating all the volumes of the samples into equal amounts of 5 ml. So now how do you determine the concentration? The concentration has to be done with a plot you do with the 0, that is a buffer which you have already subtracted, and then different concentration, then you plot, and then with the unknown you see what is the optical density or the absorbance, and from the graph you can read the concentration of the unknown. Now, having done this, this is the basic fundamental method of the measurement of the concentration of any colored compound, whether it is a dye, drug or enzyme complex. Now comes the famous equation, that is a Beer-Lambert law. This is actually developed long ago and by two basic famous scientists, Max Beer and John L. Lambert. And this I would like you to appreciate and give him credit to these scientists, which you routinely used in everyday experiments where you determine the concentration. Because the concentration, they have developed an equation. What does it say is that it has to have some limit, it has to follow the equation where the concentration of the substance you are measuring should not have any errors. Now, what does this basically law says is when the concentration is expressed in moles per liter, and when the cell, that is a cuvette you are measuring, which is the length of the cell, that means the path length where the sample is going through in that concentration is expressed in centimeter. For example, you have 1 centimeter path length, that means the light that is passing through the cuvette is 1 centimeter and your concentration of the sample is expressed in moles. Then we call the absorptivity of the molecule as the molar absorptivity. That means it is given as epsilon, and you can express the concentration in percentage also, grams per 100 ml. Then you give the molar absorptivity, the extinction coefficient, you call it as a 1%. So this is particularly constant for any molecule. That means when a molecule has its own natural absorption, that is given as expressed in the terms of the absorption coefficient E epsilon, either molar or percentage wise. So the absorptivity is, what is it dependent on? It has absorption efficiency of the sample and scattering losses as the light passes through the sample, and reflection losses as the light strikes the cell interface. So these corrections have to be taken care. So this is the one that when intensity is I0 and the light L is a path length and molar extinction coefficient is epsilon, then C is the concentration of your unknown sample which you can determine. Now it is very important, it's a very easy equation. However, it is important to mention here that in the theory what you are measuring is absorbance. So absorbance, as you know, it cannot be measured. So what you are measuring is a transmittance, which is called the minus log 10 to the base T, that is a transmittance. Now this absorbance, you write it as, what is transmittance? That is the transmittance IT by the total incident light. That is the intensity of the transmitted by the total. That is called the capital T, that is a transmittance. So minus log 10 IT by I0. Therefore, if you remove the minus, then it becomes the reversal. A equal to log base to the 10 intensity I0 by IT. So this is what you can, for example, if you don't put any sample, it has no absorption. Then what happens? All the light goes inside and it is no absorption. So theoretically all the light should come out. That means IT becomes I0. Then absorption will be 1 because log 10 is 1. I0 by IT equals IT, I0 by I0, and therefore it becomes 1. So log 1, there is no transmittance. So the absorption is 0. For example, I have shown one spectrum that we can have different samples, that is absorbance of a different, can have different wavelengths, and you can plot the equation, plot the absorption as a change in the wavelength. Now here the absorption is proportional to path length, because if you increase the path length, then of course more light will go into the sample, so the more sample can absorb. So the fundamental law Lambert proposed that absorption A is proportional to the path length of the absorbing medium. It could be a liquid, a solution, or it could be a solid also, but we are talking about in biochemical terms, biological terms, we mostly talk about the aqueous solutions or the solutions, but you can also applicable to any solids. Now the second equation, Beer's law, what it says is that you may have a length, increase the length, but if the concentration is low, if two solutions are there, one is a concentration low, one is a higher concentration, the absorbance what you measure is actually proportional to concentration. So if you combine both of them, the absorbance is proportional to concentration and the length or the distance that the light passes through the sample or the cuvette. So if you combine them, A equal to ECl, that is E is the molar excitation coefficient I just mentioned, which is a constant for a particular substance based on the concentration expressed in moles, and L becomes 1. So if you change the path length accordingly, you should remember that the absorbance will be different. Now every law has its own limitations, its own applications. So now what is the validity of this Beer-Lambert law? That there is an attenuation that has to act independently of each other. That means the medium has to be homogeneous, and also the medium that should not absorb or scatter by itself. If the medium, that means the solvent you are in which you are dissolving is giving out radiation or absorbing radiation, then what you are measuring is not the correct one. That is why when we do, when I told that we have to correct for the blank, what you are doing is to the turbidity or the scattering which you are subtracting and taking care. So that is very important. Otherwise your concentration will have, you will be expressing in a increased concentration, but which is actually not. So you are measuring, your measurement is low, but you are thinking that it is a higher concentration. Also the incident radiation must consist of parallel rays. So this part actually I will be discussing in UV absorption spectrophotometers. Different types of spectrophotometers are there. So when the light is going through the blank and you remove this blank and you introduce the cuvette in a sample. So what you are assuming is, in both cases, the light that is coming from the source is uniform. So correction is assuming that the same amount of light is actually passing through the sample as well as that. But of course, each source has its own this thing. It cannot 100% give the rays or the light in exactly same fashion. So that is one limitation. That can be corrected, that is actually corrected by the double beam spectrophotometer, which we'll discuss later. And the fifth one is the incident radiation should preferably monochromatic. It should not have the light, as you know, that it has a sigmoidal curve. That means the light should have only one wavelength and not any other radiation with different wavelength, because then the molecule can absorb in that range also and can give a different transmittance, and therefore your absorption is not the correct one. The last one is the incident flux must not influence atoms or molecules. It should only act as a non-invasive probe. So the light itself should not create any chemical reaction in the molecule. Now, having said these are important factors, even then it has its own limitations. One has to remember, mostly the students make a mistake that you take a very high concentration where the Lambert-Beer law does not hold good, but you still determine the concentration. What you are doing is that the concentration you are measuring is not correct because you are estimating it too high, which is not true. So it has its own limitation. You should do low concentrations of the substance. Now the reason is that at high concentrations, the distance you are measuring in the cuvette which is kept, the sample which is kept, when it is too high concentration, the light that is falling on the sample will not reach up to the end of the cuvette. That means the whole concentration sample which you kept in the path length, in the path of the lamp or the light, will not reach completely. It may be half of it, and then the light is over, so the rest of the molecules are not absorbing. That is one problem. The next is the charge distribution is affected by the proximity of the neighbor, and therefore alters its ability to the molecules to absorb a given wavelength. That is number 2. Now, whenever you take any analyte which is dissociating or associating, then it reacts with the solvent. That's what I told, that solvent has to be in such a way that it should not react, so that it doesn't give any, the Lambert-Beer's law in such solution does not apply or it cannot be valid. Therefore, having a different absorption spectrum for a one analyte, actually in under colored complex can give ions. So if it is one ion with colored and one with color, one molecule with color and one molecule without two molecules, one with charged and one with uncharged, both will contribute and your concentration estimation will have errors. The reaction also can cause color to the pH dependent. The solution should have a buffer to eliminate the effect. So that actually now the limitations and the validity of the Lambert-Beer's law, we keep using it so routinely that this is important to determine, to know how to derive the equation and what are its limitations. Now I'll move on to the third part of the talk, that is the UV visible spectroscopy. The term spectrophotometer is the technique or the instrument, and the principle that is used is a UV visible spectroscopy. So now major use of the spectroscopy is actually quantitative determination like colorimetry. It could be, but advantage is that you can go into UV range also, and in the visible range it gives a better sensitivity and a better you can determine the lower concentrations where you cannot, the colorimetry cannot be used. So analysis of the spectral lines also can be done. What does it mean? Analysis of the spectral lines means actually you are studying the molecular structure. That's very important, and the molecular structure actually associated with the UV absorption spectroscopy, where we will be doing it on proteins and nucleic acids. Now the atomic spectroscopy, as I mentioned, is absorption or emission of the light of the electron molecules, but then the emission we are not studying, which is a fluorescence spectroscopy. We will be dealing with it in the later classes before doing the electron microscopy. So the mass spectroscopy usually gives better results, but optical microscopy is the easiest one, and one can quickly do it to get some information about the molecules. So now what are the biochemical applications? Let us see that if you are going to do a non-polar to polar solvent, then what happens? Like if you have non-polar solvent, for example, partially you can say alcohol as compared to water, then there may be pipette transitions, and hypsochromicity can be studied. Hypsochromicity is that a spectrum moves from to the blue shift, that is the lower wavelength, or a bathochromic shift, that means the spectrum what you have shown, it moves to the right side, that is a red shift. So these terms are typically used all throughout spectroscopy, especially in the UV visible absorption spectroscopy, CD spectroscopy, and also fluorescence spectroscopy. Now the wavelength of the pi electron transition actually depends on the size and the delocalization of electrons. The pi itself, as you know, pi electrons are delocalized. So the more they delocalize, the better will be, or the easy will be the absorption. Now why I am talking about this, that we will be doing in the protein absorption spectra. There you would know that actually what is a pipette transition and where will they come. So as when we are talking about the protein, you have a peptide bond, and the protein is a polypeptide. So there are number of peptides. So each peptide has one pi-pi transition which absorbs at 190 nanometer. So you will have a huge peak or high absorption at 190 nanometer. Similarly in DNA you have an absorption at 260 nanometer. When you take hemoglobin, it is a colored compound, it absorbs at 400 nanometer. So this is just the gist. Now advantages of this is, as I told, pipette transitions can be studied, which you can use for structural analysis, conformational changes. Also you can study pi sigma star transitions, which is around 180 nanometers, and then pi-pi transition which occurs around 260 nanometers also. If the compound does not absorb between 200 to 800, then what does it mean? That it does not have a aldehyde group or a keto group or any double bond group. Then of course amino acid backbone, as I told, that is a peptide, gives around 220 to 230 nanometer, and all the side chains The side chains, because they are much more weaker, then they absorb around 250, greater than 250 nanometers. Now coming specifically to the histidine, which absorbs around 210 and cysteine around 250. Remember that these don't have high absorption as compared to other aromatic amino acids. Aromatic amino acids, as the name suggests, the pi-pi transition electronic distribution is much more delocalized. For example, tyrosine and tryptophan, they absorb around 280 nanometers, and the nucleic acids, they absorb at 260.